Morning, students. Am I audible?
Yes, ma'am, you're audible.
Yes, ma'am.
Fine. So most of you have joined, and if you are left to join, so I think we can start the lecture. So yesterday we discussed about virus transmission in human, and there are different methods for that we discussed. So can quickly just one method and example quickly. Yes, Ayushi, just one method and example.
What, ma'am?
Virus transmission methods in humans we discussed yesterday. So just quickly one method of a virus transmission with example of virus which is transmitted by that way, right? I asked Ayushi first, right?
Yes,
Ayushi, yes, switch on
yes,
your
ma'am.
camera
One
and
of the mode for virus transmission is direct contact.
Mhm.
So viruses can be spread through direct contact with an infected person. For example, the human papilloma virus, it can be spread via direct contact with the skin.
Yes, fine. Yes, the next can we have? Yes, Devanshi, next method.
Ma'am, another will be body fluid.
Mm-hmm.
Example is HIV virus, which can be spread via semen or blood.
Yes, yes. Then next, who will answer the next method? Hurry up quickly. Okay, Yuvraj.
Next method is transmission via aerosols or sneezing and coughing. The
Mm-hmm.
example is SARS-CoV.
SARS-CoV, right, right. Next method, anyone? Yes, Sam.
Ma'am, insect vectors and
Yes,
dengue and Japanese encephalitis.
yes, they all are transmitted by the insect vectors. One more left. Anyone? Yes, Manaswini.
Ma'am, am I audible?
Yeah, you're audible, Manaswini.
Okay, so one more is by contaminated food or water, where the virus will enter the body when a person, let's say they eat contaminated food or water.
Yeah,
An example of this can be the norovirus.
yeah, fine. So yes, we discussed. See, so we have discussed the transmission of viruses in plants, transmission of viruses in human or animals, which is usually common. So yes, the major difference. Yes, can anyone tell me the major difference between plant virus transmission and a human virus transmission, or a similarity? Did you notice something? Yes, can anyone?
Similarities, they both are get transmitted by the vector, like
Yeah,
in
they both have. Yes,
dengue in humans and
yeah. Yeah, that's right, Yuvraj. The similarity is that vectors such as insect vectors, they are transmitting even plant viruses also and human viruses also. And yes, insects, when we talk, the mosquitoes are the major vectors which are transmitting human viruses. And when we talk about the plants, then there are numerous group of insects basically which transmit viruses. They could be white flies, they could be aphids, they could be leaf hoppers, they could be mites also, right? So there are numerous insects in a different categories or a different group. They are transmitting plant viruses, right? So today's lecture, we are starting with the life cycle now, and today we'll discuss about the life cycle. I'm starting with a very general one. So let's first discuss the life cycle of a tobacco mosaic virus, a simplest one to explain and understand, and then we can go to the life cycle of a viruses when they infect prokaryotes, right? When they infect bacteria, so what is their life cycle? How they do this? Lytic and there is a lysogenic life cycle, and then we'll go to the next lectures. We'll go to the life cycle of a viruses when they infect humans, and then we can at the end we can find out the major differences, that at what stages they are different, at what stages how they are different from the plant, different from the humans or prokaryotes, how what is the major difference, so that we can go. So today I thought let's start with the plants, the most basic one and the most general one, that uh, very easy to understand, right? So viral infection, we know it can be asymptomatic, or it can lead to cell death, could be a latent infection, right? Latent infection, that means it is asymptomatic even when we talk about it in plants. So it is not causing much losses. It is there in the plant, but not much symptoms are being produced. So we call it as a latent infection, right? Many viruses, they are latent. For example, if we talk about viruses in stone fruits or pome fruits. So pome fruits may majorly it's the apple, and the stone fruits is the plum, peach, apricot. These all they come under the category of a stone fruits. They have a stony endocarp, right? So in those cases, there are few viruses which are latent. They are always there. They might not be any symptom on the leaves, but when we just try to isolate RNA or mostly they have RNA viruses. Yes, one DNA virus has been reported on the apple recently from China, Vego virus, but otherwise they all are RNA viruses, and there are viroids which are being reported. So apple chromatic leaf spot virus, apple stem fitting, grooving, these are the all different viruses which are latent there. We will not find any symptoms, but still if we try to isolate RNA and then we go for a PCR for a detection or by ELISA, we'll see that the virus is still present, right? Or they can lead to cell death. That is a lytic infection. Definitely this light, if it is killing the cells, so that means the infection is a lytic, or lytic life cycle is being followed. The life cycle, basically how it start with the penetration of the virus into the host cell. It enters. The first step is it is penetrating. Then the virus is uncoated within the cytoplasm of the cell. When the capsid is being uncoated, capsid is removed. Then depending upon the nucleic acid, cellular components are used to replicate viral genome, and then further viral proteins are being synthesized for the assembly of the new virions or a new whole virus particle. So to establish a systemic infection, then definitely the virus needs to enter a part of the vascular system of the plant, such as phloem. It must reach that point. The time required for systemic infection, it may vary from few days to few weeks. It totally depends upon the virus, the plant species, and the environmental condition. Then the virus life cycle is complete. When it is considered complete, when it is transmitted from infected plant to the healthy plant. Further transmission is taking place. That means the life cycle is completing. So if you just look at the graphical view of a viral life cycle or how virus multiplies. So it is basically first inoculation. Inoculum is basically, what is inoculum? What do you understand by inoculum? Inoculum is the infective material which has a virus and it can cause a disease. It's called an inoculum, right? It could be anything, any starting material, right? So inoculum is inoculum of virus. It is entering, it is binding to the cells some way, it is entering, and then after that the second stage is called the eclipse stage. The eclipse stage is a stage when the virus, it is penetrating the cells, right? At that stage nothing is happening, it is penetrating, it is preparing for the replication and further multiplication using host cell machinery. So for some time nothing happens. So that stage is eclipse stage, and then the host cell releases virus particles. So burst stage it is called. So that means the virus particles are being formed now and they are being released. Then the fourth stage is a burst size. That means the number of virions released per bacterium. So this is the example given of basically a bacteriophage, that it is infecting a bacteria. So what is the pattern that is being followed by the virus in the multiplication cycle or a life cycle, right? So four main stages are there when we talk about tobacco mosaic virus. I'm taking an example, a simplest one, the first virus which is being identified. So these first stage is a transmission of virus particles. Second stage is an infection. Third stage is a virus multiplication. And fourth is the sites of viral RNA and viral protein synthesis, how it is being taking place in the host cells. So transmission of virus particles, we know the tobacco mosaic virus is a rod-shaped particle. It infects leaves of the tobacco plant and it is transmitted, and complete particles, they are introduced into the host cell by the insect vector. So tobacco mosaic virus is transmitted by mysis species, aphid species, and some species of the grasshoppers also. The virus is also transmitted mechanical means. So mechanical means if we are doing it in a lab, that means we can just take a inoculum of the virus or a plant extract from an infected. We know that that particular plant is infected by this particular virus. So that extract in a particular extraction buffer will take and will rub it on the virus leaves, and then we are doing it directly. That means a mechanical transmission that we are doing, or in the field it can be done by the using infected uh, equipment such as shatiyars, or by the human clothing also. If there's abrasion on the plant, it can be done, right? So virus is very contagious, and the person who has handled cigarettes containing infected tobacco leaves may transmit the virus to the healthy plants if with bare-handed he's doing all this process. So there are chances that virus through his hands also the virus can get transmitted. Then the second stage which we talk about is the infection. So infection takes place with the whole virus particle, and separately the virus components are not infectious. So how we know this? There are some studies which are being done by the people that they separated the protein code from the RNA code by using certain chemical procedures, and they found that the protein code alone is not capable of producing infection. The RNA is alone is infectious, but much less than that of the intact virus particle. So this is the older study. These days, how do we do these studies? We produce, we synthesize or we construct infectious clones. Usually these kind of studies are being done in the viruses, where there are satellite virus particles also. They have DNA components, and then they have a certain satellite particles such as beta satellites or alpha satellites which are being associated with them. So researchers, they wanted to know whether these satellite particles, they play some role in the infection or not. So how did they study that? They prepared infectious clones. So they artificially, or not artificially, they took the DNA of a particular virus, virus that they want to study, and they put it in a some vector, and then they transplant or they infected using agrobacterium. Transformation was done and plant में वो उसको introduce किया so that they can see the symptom. Then second, they did only the alpha satellite or a beta satellite. They made its infectious clone. So, and then they transformed it again into the plant. And then they waited till the symptoms. They found that the symptoms were developed only in case of singly when the genome DNA, only DNA and few genes are there. There's a code protein gene, there's a movement protein gene, then there are other genes. But satellite particles alone cannot cause any symptoms. So that means they do not have any particular role there. But when in combination with the DNA component, they are infected in the plant, and when we see the symptom, it leads to the enhancement of the symptoms. So by such comparison studies, we can see that which parts in the viruses are playing a role in the symptom development. So these studies are being done to prove that which like they did some study. They found that code protein alone is not capable of producing the infection, whereas RNA is infectious, but still the intact virus particle is more infectious as compared to the individual one. So these kind of studies are being done. So separately, they found both viral RNA and protein are readily attacked by enzyme in the host cell and are decomposed. Singly, RNA is if also there, definitely we know that it is quickly destroyed by the ribonuclease enzyme which is in the host cell. So virus particle, when they are intact, they are very resistant to decomposition and they are more stable and viable. For infection, wounding of the host cell wall is necessary to produce infectable cells. So how virus will enter, there need to be some way. So wounding, if it is through the vectors, then vectors, they feed on the plant, wounding is being done, and then if they probe, they introduce the virus into the plant. But if mechanical transmission we are talking about, then we need to do certain kind of abrasions in the plant's outer epidermis and cell wall basically, and there is a carborundum powder basically which is used. So very lightly we rub the leaves with that carborundum powder, and then we just inoculate the material, or it is caused by the mouth parts of the insect vector or wounding epidermal cell or breaking epidermal hairs by abrasions, right? So through these infectable sites, the virus particles, they gain entry into the host, and then they come in contact with the host cell membrane. So by the process of pinocytosis, the virions, they enter the host cell protoplast. Now, stage 3 is the virus multiplication. So after infection, the virus particles, they disappear from the view. It ceases to exist as an organized unit and then break up into its two component parts in the host cytoplasm. The viral RNA is released from the protein coat. So uncoating of viral RNA is the initial steps towards the virus multiplication. Now it takes place within the minutes after virion, they get entry into the host cell. This stage is known as the eclipse stage. That means it is virus has got entry and now for some time nothing happens. And thereafter it starts the multiplication process. So viral coat protein remain in the host cell cytoplasm. The RNA component, they move from cell to cell, causing the production of new virus components, which then assemble to form more virus particles. So these are the ACO et al. In 1966, it reported that virions of wheat yellow virus, they move intact from cell to cell through plasmodesmata. So if we study in your course, you don't have the movement of viruses. So when we study the movement of viruses in the plant, so there's a proper mechanism that how through plasmodesmata the viruses are moving and how how they change the size exclusion limit of the plasmodesmata, basically how the viruses, they change the SEL, size exclusion limit of the plasmodata, and then they gain the entry inside that, right? So step 4 is the site of viral RNA and protein synthesis. So most widely held view is that virus protein and virus RNA are formed by two separate systems. So when they enter the host cell, viral RNA, it takes control of the host cell machinery. So it uses it to produce the viral component instead of the host cell parts. The viral components are then assembled into a new virus particles. The viral nucleic acid, it combines with or replaces part of that with the host cell also. So thereafter, the host cell produces viral components at the expense of its own. The other commonly held view is that DNA of the host cell uses viral RNA to elaborate more viral RNA and a protein. So they are elaborated in the cytoplasm of the host cell, while the viral RNA and then first they do the viral RNA and then the viral protein. So it is difficult to understand that why host plant DNA, which is coded with specific information to build plant RNA and the plant protein, and it start building the viral components also. So there is also a suggestion that ribosomes in the cytoplasm of the host cell, they are associated with the elaboration of the viral component. And this is based on the fact that viral RNA is somewhere related to the ribosome RNA. So entering the host cell, viral RNA, it overrides that of a ribosomes and it uses the ribosome to produce the viral components. The ribosomes, however, are considered to have no genetic continuity, so it is very difficult to associate them with the replication of viral specific RNA. So some virologists, they say that replication of viral RNA with the chloroplast of the host cell also, but not much evidences are being there. So the biochemical, cytological, and genetic data have proved that chloroplasts are semi-autonomous self-duplicating organelles of the eukaryotic cell. So that is the basis of this theory, that they believe that they could be using the chloroplast. So they thus have a genetic continuity of their own, and they contain RNA and DNA distinct from nuclear constituents. So the replication of chloroplast DNA takes place within the particular organelle. So it is possible that DNA of a chloroplast, they codes for the replication of viral RNA and protein also. The widely accepted view is that viral components are formed by two separate systems. The released viral RNA, it reaches the nucleus and there it takes control of the host cell machinery. So it uses the host cell machinery to produce the viral component instead of the host cell parts. The nucleolus in the nucleus produces the new viral RNA, which later escapes to the cytoplasm around the nucleus through channels present in the nuclear membrane. The tobacco mosaic virus protein is synthesized in the cytoplasm, which is very near to nucleus on the cytoplasmic ribosomes. The assembly of new tobacco mosaic virus particles, again, it takes place in cytoplasm on endoplasmic reticulum. And the eclipse stage, which lasts for around 6 hours, right? It takes around 6 hours to do that. And by that time, the first progeny of tobacco mosaic virus, it appears in the cytoplasm. However, the result of experiment with fluorescent microscopy and some other labeled antibodies and investigation, they tell a different tale. They are at variance with the majority view. They indicate that tobacco mosaic virus, protein and RNA both are synthesized within the host cell nucleus. The assembly of TMV particles, they also occurs in the nucleus. The assembled TMV virions of a particle, then they escape in the host cell cytoplasm where they accumulate. Now the attack of TMV causes leaves to wrinkle. When they increases in number, it causes leaves to wrinkle and they become mottled. Now mottled effect of a leaves, they gives like the appearance of a mosaic. The disease thus came to be known as a tobacco mosaic disease or a tobacco mosaic virus in the tobacco. So see, the point here is the first when I first studied about the tobacco mosaic virus, the first thing that came to my mind was that why we are studying tobacco viruses? Isn't it that tobacco is harmful? Why actually we are bothered if tobacco there's some virus which is affecting tobacco plants? The first thing was this in my mind. But then when I just went very deep into when we this was when I started studying virology in my master's program. It was virology was just a course, but when during my PhD I did started working on the research, rather a research work on the viruses, and then I came to know that tobacco is a big industry. It is giving employment to a lot of people throughout the world. So that's why the viruses, though they are not much losses or in the content, the virus is not affecting any, they are just affecting the leaves. They are making them crinkle. Yes, could be that the taste of tobacco at the later stages could be affected by the virus, but otherwise it is not affected. But then another point was that tobacco mosaic virus is not only affecting tobacco, it is affecting other vegetables or other crops also. So this is why, that was why it was important to study this virus, and not just because it is affecting tobacco and tobacco use is, though it always comes with a warning that tobacco use is injurious to health, but still it is being sold at every point and every place, maybe outside the universities, outside the hospitals, and everywhere in the shops you'll find this, right? Though it always comes with a warning, still people use that, right? So that was another story. But this is how initially the work started. That initially they did not have any methods to determine that how virus is replicating. At this stage, if we talk about the various methods, there are a number of methods to study basically life cycle of a virus. We can do it. Crystallography is a study. Basically, we can study about the virus structures and all those things. Then there are other methods. Electron microscopy gives us the view of the virus particles in general, right? So at various stages, how the life cycle is going on by observing those stages under the electron microscope and other methods which are available. We can just it gives us a view of that how it is actually multiplying and how it is developing. So this was the simplest virus life cycle, like virus, which basically, yes, did someone left or entered? I guess, I don't know. All of a sudden I saw someone. Okay, so this was the basic, just the simpler process, that how it is penetrating, how it is entering, how it is multiplying, and how it is again a whole virus particle is formed and they're releasing and they're going and infecting the other plants. So this is how a life cycle of a virus in a plant is being completed, right? So this is all for today, and in the next life cycle of a virus, which is on the prokaryotes, which is even more interesting, where basically which is about the lytic life cycle and a lysogenic life cycle. So virus can either kill the cells and then move ahead, or it just multiplies with the go and attach the genome of the host and multiplies with the host, and okay, there's some sort of a collaboration kind of going on, that I'm not going to kill you, but yes, I'm there with your genome and I'm multiplying with your genome. So we'll try to discuss those cycles in the details. If there's any question, please ask your questions. And in the assignment, I have just posted one question, which is a very general. It is in the Microsoft form only. So all of you, please just try to do that question within 5 minutes and then come back so that we can discuss also, right? Let me please. Is it visible? No?
No, ma'am, it's not visual.
No. Just a moment. Assignments. Okay, students, your FBL class is not going on.
No, ma'am.
मैम एक्चुअली कल सर का फोन आया था, सनी सर का, तो वो कह रहे थे इशू उन्होंने करा था, पर उन्होंने मेल भी करी है, बट अभी तक किसी का रिस्पांस नहीं आया है।
लेकिन मुझे लगता है कि आप लोगों को एटलीस्ट बताना चाहिए था, बिकॉज व्हाट वी थॉट वाज कि आपकी क्लासेस जो हैं वो they'll be somewhere direct, like मतलब
मैम मैंने स्टार्टिंग में सर को बताया था, मैंने मेल भी सर को कल भी करी है मेल।
Okay, just a moment, I have to check my assignment where it is called. Okay, assignments. Just a moment. Where is the assignment from? Okay, should be visible now, I guess. Is it visible?
Yes, ma'am, it's visible.
Yeah, I just publishing why this mic is not working. All of you, please quickly do it and then come back.
Ma'am, I actually had a doubt with today's session. So, ma'am, you know how it's mentioned that the viral RNA and proteins, if they, if they are separate in the cell, then it's immediately attacked by the enzymes of the host cell, right? So, how come in the infection stage, they're separating and the virus is able to replicate and all?
No, I didn't get your question, Manas.
Okay.
See, viruses. See, when inside the cytoplasm, viruses they uncoat themselves, right? So when they uncoat themselves, then by using the host cell machinery, they are forming the further virus proteins or virus cell parts, basically, right? So they say when they enter at the entry stage, when they enter, if they are if they are entering in a single wave, they are not intact virus particles, then there are chances that the enzymes of the host cell enzymes will degrade them. But if inside the cytoplasm they are going and uncoating, then they will not affect them.
Oh, okay, ma'am.
So at the entry stages, when they are entering, right, when something from the outside is just penetrating, just at that stage, there are chances that those enzymes will directly degrade them if they are not intact. So they're going inside, and then they are uncoating and doing this process. So then they are not affecting them inside the cytoplasm, right?
Okay, ma'am. Thank you.
Hello. Yeah, I'm in a class. Will call you.
Ma'am, do we have to uh, write the steps or we have to explain each step?
No. You just write the steps and just 2 lines each step, not more than that. Because in that form, you cannot explain in detail.
Okay.
Yes, done. Yes, please start submitting. Yes, students, please submit. I still see 1, 2, 3, 4, 5, 6, 7, 8, 9. Just give me a minute. Hello. Yes, most of them submitted, but still few are not. Okay, in the meanwhile, I'll start taking attendance. Also, please switch on your cameras. Excuse me. Adhish Mishra. Adhish. Yeah, am I audible? Adhish Mishra. Athoiba.
Present, ma'am.
Athoiba.
Am I visible?
Yeah, yeah, your camera is on, but I can't see you. It could be the network issue. No worries. Yeah, you're visible now. Yes, others, Athoiba, please switch on your camera. Ayushi, others also, please quickly switch on your cameras. Ayushi,
Present,
Sam,
ma'am.
Sam, Devanshi,
Present, ma'am.
Ishita, excuse me, Janvi,
Present, ma'am.
Kshitij,
Present, ma'am.
yeah, Manaswani,
Present, ma'am.
yeah, Namrata,
Present, ma'am.
Nandini,
Present, ma'am.
Nehal,
Present, ma'am.
excuse me, and Rupal,
Present, ma'am.
Sahiba,
Present, ma'am.
Sanjana Sharma, Shakti,
Present, ma'am.
Shivani Mishra, Tanvi,
Present, ma'am.
Present, ma'am.
Hridev,
Present, ma'am.
Vivek, Yukta,
Present, ma'am.
Yuvraj,
Present, ma'am.
Ishita has not turned in, and Athoiba, Adish, and Ayushi, you have not submitted, right? You have not. Sahiba, you have also not. Shivani, you have also not. Yuvraj and Vivek, and they have also not submitted, right?
Yes, ma'am. I'm just
I'm
uploading.
Yeah, upload, please. Sanjana Sharma, present. Those who have uploaded can leave the meeting. Others, please hurry up to it.
Thank you, ma'am.